Bismillahirrahmanirrahim. Today I will discuss coagulation cascade, the classical blood coagulation pathway. This is a second lecture of hemostasis series. In previous lecture we discussed platelet plug. So coagulation factors are protein in nature, and they are present in blood in inactive form. They are non-active. They help in the formation of clot, and this clot actually then stop the bleeding. That's why they are called clotting factor. They are also called clotting factors. There are several clotting factors in the blood, and when there is deficiency or when one of them is leaking or missing, it can cause clotting disorder or bleeding disorders. They are where they are actually synthesized. Most of the coagulant or procoagulant or clotting or non-clotting factors are actually synthesized in the liver, except these factors. One is the tissue factor, which is also called thromboplastin, and calcium, which is factor 4, and factor 8, which is called antihemophilic factor. Antihemophilic factor is very important because when this factor is missing, a person bleed heavily, and it doesn't stop without support. These proteins undergo, when they are activated, when they undergo under the influence or with the presence of vitamin K, gamma carboxylation of glutamic acid. They are vitamin K dependent gamma carboxylation of the glutamic acid. When carboxyl group is attached to these proteins, they are activated. They produce a point or a space on their surface to which calcium is attached, and then it precipitate then it participate in the clotting cascade. Deficiency of vitamin K or administration of vitamin K antagonist warfarin. Warfarin is vitamin K antagonist, lead to anticoagulation. Actually, this warfarin nowadays drugs are available like clopidogrel. They are cyclooxygenase inhibitor, and they are used in the coronary heart disease and other coagulative abnormality. But still warfarin is also used for prevention of coagulation, especially in coronary heart disease. And warfarin is anti-K or potassium, and this prevents carboxylation of the glutamic acid residues. Now, numerical nomenclature of coagulation proteins. Nomenclature of coagulation protein is rather complex. The first 4 of the 12 originally identified factors are referred to by their common names. What are their common names? Factor 3 is also called tissue factor or tissue thromboplastin. Fibrinogen is also called factor 1, and prothrombin is called factor 2, and calcium is factor 4, and also assigned Roman nomenclature. So they are called by their common name, tissue factor or fibrinogen or prothrombin, or they are called factor 3, factor 1 or factor 2. So they have both the Roman nomenclature and also they have their common names. Factor 6 actually doesn't exist or yet not are discovered, or any factor is given this nomenclature. The most recently discovered clotting factors, for example, prekallikrein and high molecular weight kininogen, have not been assigned Roman nomenclature. So these factors are called with their original names. Some factors have more than one name. Like factor 3, it is named tissue factor or tissue thromboplastin. Factor 5 and factor 8 are also referred to as the labile factor because their coagulation activity is not durable in stored blood. When you store blood and when a coagulation process, when this blood is transfused, so coagulation process in that blood is not very active. So that was why it is these factors are called labile factor. They are not, their action is not pronounced in stored blood. So these are the factors and their name. So fibrinogen is factor 1, prothrombin is factor 2, tissue factor, it is also called factor 3, or tissue thromboplastin, calcium, it is given the Roman nomenclature. 4, factor 5 is procoagulation, factor 7, 6 is not existing, so is serum prothrombin conversion accelerator, and factor 8, it is antihemophilic factor. Very important, as I told you that if a person is lacking this factor, he is prone to bleeding. Factor 9, it is called plasma thromboplastin, and factor 10, Stewart factor. Factor 11 is called plasma thromboplastin. Factor 8, Hagman factor, and factor this is factor 12, and factor 13, fibrin stabilizing factor. So these factors are either named given the nomenclature of Roman Roman nomenclature, or they are named with the scientists who discover them, or they are named according to their function. So they are also included in these factors. Prekallikrein, it is called pleasure factor, high molecular weight factors are kyanogen, are high molecular weight, kyanogen and platelets. So they are include. These are the 13 factors, and these three, they are actually active factor which take place in the coagulation process. As I told you that some of the factors are given the Roman nomenclature, and when they are non-active, they are written in the Roman nomenclature. But when they are activated, so A is appended or added to it. So this A actually shows that the factor is in active form. Now, downstream activity of coagulation factors. The coagulation factors are generally enzymes called serine protease. These are enzyme in nature and they are called serine protease. Serine protease are enzymes that cleave peptide bond in protein, in which serine serve as the nucleophilic amino acid at the active site. So they actually they are neutrophilic to amino acid. They attack the amino acid and break down them, which act by cleaving down protein. The exception are tissue factor, factor 5, and factor 8, and factor 13. This serine protease does not act on these factors. They are excluded from that series. Tissue factor, factor 5 and factor 8 are glycoprotein. These two factors are glycoprotein in nature. We know that, protein with sugars. And factor 13 is a transglutaminase, class of transglutaminase. Transglutaminase is class of enzymes that catalyze the formation of covalent bond between the gamma carboxamide group of protein. It actually causes carboxylation of the protein. Now, coagulation pathway. The coagulation cascade is classically divided into three pathways: the tissue factor, contact activation pathway, and both this tissue factor and contact activation pathway finally activate a common pathway, factor 10, which is called thrombin, which convert thrombin into fibrin. So, from activation of factor 10, the third pathway actually started. Now, tissue factor pathway is also called extrinsic pathway. The main role of the tissue factor in this pathway is to generate thrombin burst. When there is tissue damage, this tissue factor or factor 3 is released and it actually generates thrombin burst, which means that they activate or produce thrombin in large quantity, and which is the thrombin is the important constituent of the coagulation cascade. So, when there is injury, large amount this thrombin burst is activated and large number of thrombin is released. Following damage to the blood vessel, factor 7 leave the circulation and come in contact with tissue factor expressed on tissue. Factor bearing cells, stromal, fibroblast, and leukocyte. When there is damage to the tissue, these cells actually expose the fibroblast or collagen tissue, and leukocytes are exposed. And this actually attract the factor 7, and the factor 7 leave the plasma and attach to these cells which are present on the damaged tissue surface, forming an activated complex, tissue factor and activated factor 7. Tissue factor are released by the damage, and when with this tissue factor 7 is attached, it is activated, and they make a complex which is called tissue factor and factor 7 complex. This complex activate factor 10. When this complex is formed, it acts on factor 10 and activate it. A is added, appended to it. It means that this is activated factor 10. Factor 10 and its cofactor 5 form the prothrombinase complex. When they combine with each other, they make the 10 and 5 make prothrombinase complex, which activate prothrombin to thrombin. Thrombin then activate other component of the coagulant cascade, including factor 5, factor 8, which form a complex with factor 11 and activate and release factor 8 from being bound to 1 Willebrand factor. 1 Willebrand factor, they are then attached to the 1 Willebrand factor. Factor 8, activated factor 8 is the cofactor for activation of factor 9, and together this factor 8 and 9, when they combine, they form the kinase complex which activate factor 10. Kinase means 10, and they are given the name kinase because they are activator of the factor 10, and so the cycle continues. The contact activity pathway. This is called intrinsic pathway. In the previously we as discussed that it starts from the tissue factor and end on the activation of factor 10, and then the formation of prothrombin to thrombin, and then ultimately synthesis of fibrin. The contact activation pathway began with formation of the primary complex on collagen by high molecular weight chironogen, prekallikrein, and factor 12, Hagman factor. Factor 12 is also called Hagman factor. Prekallikrein is converted to kallikrein, and factor 12 become activated factor 12. When this reaction occurs, factor 12 is also activated, and activated factor 12 convert factor 11 to activated factor 11. Now activated factor 11 activate factor 9. This is 9 and this is activated in the activated form, which with its cofactor 8 activated 8 from the kinase complex, which activate factor 10 to activated factor 10. So in this complex, in this cascade, you can see that in the extrinsic pathway, it also end at factor 10, and the intrinsic pathway also end at factor 10, or they are both the pathway actually activate factor 10. The common pathway scheme implies that prothrombin is converted to thrombin only when activated upon by the intrinsic or extrinsic pathway, which is an oversimplification. It means that factor 10 is either activated by extrinsic or by the intrinsic pathway. In fact, thrombin is generated by activated platelet at the initiation of the platelet plug, which in turn promote more platelet activation. Thrombin actually activate in the primary plug, we already know that it causes activation of more platelets. Now, function of thrombin. Thrombin functions not only to convert fibrinogen to fibrin, it also activates factor 8 and 5 and their inhibitor protein C in the presence of thrombomodulin that we have discussed. Thrombomodulin we have discussed in the first lecture, that factor C and this protein C is activated, and it is and it activates factor 13, which form covalent bond that cross-link the fibrin polymer that form from the activated monomers. So in this mechanism, actually fibrin clots are formed and they then deposit on the primary platelet plug and stabilize it. The coagulation cascade is maintained in a prothrombic state by the continued activation of factor 8 and factor 9 to form the kinase complex until it is down-regulated by the anticoagulant pathway. When there is injury and when this activation starts, so there is continued activations of these factors by down-regulations until when the bleeding is stopped and they are not required anymore. So at the last anticoagulant activity is started, and then the further propagation of coagulation is prevented. So these are the outcome of the pathways. In extrinsic pathway, tissue factor are damaged and they release tissue factor is released due to the damage of vessels, and the components are tissue factors 7. They actually activate the tissue factor 7 and activation of factor 8. When these two are combined, the tissue factor and factor 7 combined, they activate factor 10. In intrinsic pathway, surface contact, exposure of the collagen factor to negatively charged vascular subendothelial surface, thrombin. Thrombin is negatively charged, release at the end of the common pathway, positive feedback loop. And this pathway activate factor 12, factor 11, 9, 8 and platelets, membrane, phospholipid and calcium ion. Activation is, the end result is activation of factor 10. And common pathway, factor 10 activation by either the intrinsic or intrinsic pathway. Here activation is factor 10, factor 5, prothrombin, platelet membrane, phospholipid, calcium and fibrinogen, and generation of thrombin converts fibrinogen to fibrin, activate intrinsic pathway, activate factor 13 and platelet activation regulation of clot formation. Regulation of coagulation cascade. Without regulation mechanism, the positive feedback loop triggered by thrombin in the intrinsic pathway would continue indefinitely and causes dangerous level of clotting in the blood. So regulation is important. If it is not inhibited, when these pathways are activated, their inactivation is also important. For this reason, various protein inhibit aspect of the coagulation cascade. Once enough thrombin has been produced, protein C is produced in response to thrombin, binding to the receptor thrombomodulin, which we have discussed in the previous lecture on the vascular endothelium. This leads to protein S activation, which then weak down factor 5 and factor 8 in a negative feedback mechanism. So there is activation and there is inhibition of the coagulation cascade. Antithrombin is an unnatural anticoagulant that is produced by the liver and destroy factor 11 and 10 and thrombin. This antithrombin is actually anticoagulant and liver produces it. Heparin and pondaferinox enhance the natural effect of antithrombin in inhibition thrombin and factor activated factor 10. When heparin is given or pondaferinox is given, they actually inhibit factor 10 and thrombin. This factor pathway inhibitor is a protein that binds to and inactivate factor 7 and factor 10. So this is the actually intrinsic pathway is activated by surface contact when the vessel wall is damaged and surface contact of these coagulative factors occur. So, and the extrinsic pathway is activated by the tissue damage. When the intrinsic pathway is activated, it acts on factor 12 and convert it into activated factor 12. This activated factor 12 then act on factor 11 and convert it into activated factor 11. Activated factor 11 in presence of calcium activate the factor 9 to activated factor 9, and it need a complex of factor 8, platelet factor and calcium. So in this way, when the factor 9 act on factor 10, factor 10 is activated. Then this factor 10 act on prothrombin and it convert it into a thrombin. And when factor 8 is activated, this thrombin act on fibrinogen and convert it into fibrin. And when activated factor act on fibrin, convert it into stabilizing fibrin clot. So this was the whole history here. One thing is important, that the common pathway is activation of factor 10. It is activated when you can see that from here, when the intrinsic pathway is activated, it ultimately activates factor 10, and when the extrinsic pathway is activated, it also activates factor 10, and from here onward another pathway is started which end on stabilizing fibrin clot. This is a simple diagram that in intrinsic factor 12, first 12 is activated, then 11, then 9, then 8, and ultimately 10 is activated. Here tissue factor 7 prothrombin, and here factor 10 is activated in the presence of factor 5 calcium and lipid, platelet lipid, phospholipid. Prothrombin 2 is activated to thrombin, and thrombin actually convert fibrinogen into fibrin clot. So thank you. This was the whole history of hemostasis, the primary plug and the secondary plug, the stabilization of the platelet plug, which is formed in the primary hemostatic cascade or primary hemostatic phase. By these coagulation factor, this loose factor is then converted into a stable clot. So that is called secondary clotting. So thank you. Hoping you may have learned from this lecture. Again, request for the subscription and press the bell icon to get in time information when new lecture is uploaded to the channel. Thank you.